Today we will learn about chapter 4. Chapter 4 is about sex determination and sex-linked characteristics. So in chapter 4 is composed of three parts. First part is we will learn about sex determination, and also second part we will learn about sex-linked characteristics. Finally, we will learn about dosage compensation. So this is the reptile, and this is a reptile. And in the reptile, the sex determination is determined by like this. They have ZW system. For example, ZZ is male and ZW is female. How about the human? Human is we use the XX and XY system. So XX is female and XY is male. A little bit different, isn't it? Okay. So we will learn all of the system in detail and in the later slide. So in this slide, we will learn the sex determination. So there are several different mechanisms of sex determination. For example, in the reptile, in the ZW system, and grasshopper in the XXXO system, and also the haplodiploid system. So we will learn the XXXO and haplodiploid system in the later, in the next slide. And the human is I already mentioned we use the XY system. So this is the X chromosome, this is Y chromosome, and this is X chromosome. So they are totally different in the size, but this the Y and X chromosome are pairs during the meiosis. So this is the meiosis 1, metaphase 1. So usually, even though they are not homologous, they are X chromosome is Y chromosome, they pair each other. So even though they are not homologous, means that genes located on each chromosome are different, because around here and around here, the genes located in the more similar region is totally different. In this slide, we also learn the sex determination. So in the chapter 2, we already learned about the sexual reproduction. So usually the sexual reproduction is alternate between haploid and diploid. So usually the meiosis produces the haploid gamete. After fertilization, so produces a diploid zygote like this. So this is we already learned in chapter 2, the meiosis implant. They also use their sexual reproduction is alternate between the haploid and diploid. And also this is a meiosis in human. So usually in the male, the male specifically we call it as spermatogenesis, and also in the female we call it as specifically the oogenesis. Usually the sexual reproduction alternates between haploid and diploid status, and also most organisms have two sexual phenotypes, male and female. Male and female. So most organisms have two sexual phenotypes. We call it as a male and female. So last slide we learn about most organism has both sexes, for example male and female, but sometimes both sexes is located in the same organism. So we call it as the hermaphroditism. In Korean we call it as 암수 한몸성. And specifically, so we call it as monoecious. In Korean, we call it as 암수 한 몸의. It means that both male and female reproductive structure in the same organism. For example, this is monoecious flower. All of flower, nearly more than 90% of flower is monoecious. So in the one organism, in the same organism, is the, for example, anther. Anther is a male reproductive structure, and the ovary is also female reproductive structure. Usually in the flower, it's monoecious, means that 암수 한 몸의. But sometimes flower is the dioecious. Dioecious means that either male or female reproductive structure in one organism. For example, this is we call it as staminate flower. Staminate flower, they only have the anther. And also another example is pistillate flower. It means that this flower only has the ovary. Okay, so but the flower is nearly more than 90% is the monoecious. How about human? Human is dioecious. So female has the ovary, but our male has the sperm. Sperm as the male reproductive structure. Female has the ovary as the female reproductive structure. Okay, so in this slide we will learn about sex chromosome and non-sex chromosome. Especially we call it as a non-sex chromosome is autosome. In Korean we call it as 상염색체. And also sex chromosome we call it as a 성염색체. So this is an example of human. Human has the sex chromosome is X or Y. So female has the XX, male has the XY. But human also has autosome. Autosome means that human has 22 pairs. So totally our human has 22 pairs plus XY is the male and 22 pairs plus XX is the female. Okay, so also now we will learn about another sex determination type. One is XXXO system. Usually we can see this system in the grasshopper. Okay, so first XX shows that the female, but XO means that they only have one X chromosome. It will make the male like this. So this is the grasshopper. So grasshopper they have the autosome, 11 pairs of the autosome. So totally 11 pairs means that 22 chromosomes. Okay, when they have the X chromosome, they will become the female grasshopper. If they have only X chromosome, they will become the male grasshopper. How about human? Human and usually in the mammals. So usually in the mammals usually we use the XX and XY system. So this is one example in the human. Human is that XX is female and XY is male. So XY male, they can produce the gamete, haploid gamete X or Y. Female, or they can produce the haploid gamete XX or X. So this is a Punnett scale. So it's also after fertilization. So male, they can produce the X and Y. Female, they can produce X and X. And this scale, they will make the XX. It means that it will become the female. And also in this carrier will be also XX, and this will make the female, and this carrier they will make the XY, means that these crosses make the male. And how about XY in this carrier? They will make the XY. So it means that 1-to-1 sex ratio will be produced. Okay. Okay, so this is we show that human or mammals, including human, they will show that they will have XX and XY system. So human has the uh, 22 pairs, and also human has the XY as the sex chromosome. Okay, so we will learn the XX, XY system once again. Okay, so this is the X chromosome. So X chromosome is usually they are larger than the Y chromosome. So X and Y chromosome in human, also most of the mammalian cells, they differ in size, and also they are differ in genetic content, but they have the homologous region. So we call it as the pseudo-autosomal region. This is the pseudo-autosomal region. Uh, the, uh, the, the region located in the short arm, we call it as primary pseudo-autosomal region. And also the region which is located in the longer arm, we call it as secondary pseudo-autosomal region. So this pseudo-autosomal region is essential for XY chromosome pairing in meiosis, specifically metaphase 1. So previously, I didn't explain the why X and Y chromosome pairs during meiosis, even though they are not homologous, even though the genes located on each chromosome are different. Why? Because of they have the primary pseudo or autosomal region, and also they have secondary pseudo autosomal region. That's why they can pairs each other during meiosis, specifically metaphase 1. So in this slide, we will learn about GW system and also haplodiploid systems. So okay, in the GW system is shown in the birds and reptile and butterfly and some amphibian and fishes. For example, Z is male and ZW is female. So this one is exactly opposite to the XX and XY system in mammalians. For example, in our mammalians, including humans, XX is female and XY is male. So this is the one of the GW system which occur in birds. So this is chicken, okay? Chicken, and dad is father is Z, and mom is ZW. So dad make the gamete like the Z and G, and the mom they can make the ZW. So after this is the Punnett care. In this care, they will make the male, and this care they will make the female and females like this. Okay, so how about another the system is the haplodiploid system. Haplo and diploid system, which occurs in bees and wasp. Wasp means the marble, and also ant. So haploid set is the male, haploid N is male, and diploid is the 2N is females. and usually in the parent generation, the female, they did the meiosis, so they make the gamete in the egg, the N and N in the egg. So if there is no fertilization, and this egg will make the male, which has the haploid set and and zygote, and also, but when the fertilization happens, egg and sperm, they meet each other, so finally they will make the 2N zygote, and this one will become the females. So conclusion is in haplodiploidy, sex is determined by the number of chromosome set. For example, N set or 2N set. Okay. Okay, this is concept check. How does the heterogametic sex is differ from the homogametic sex? What is the heterogametic sex? For example, in JIT system, ZW is the heterogametic sex, and also XY is also heterogametic sex, and XO is also heterogametic sex. Okay, so usually the heterogametic sex is ZW. They are this is occurs in the female in the reptile, and also XY is male in the murmur, and XO is male in the grasshopper. But the question is the heterogametic sex is a male and the homogametic sex is a female. Is right? Is wrong. Okay, so what is the homogametic sex in the JIT system? In JIT is the JIT is the male in GW system, in XX is female, and XX is female in XXXO system. So okay, how about another criteria? Okay, another question is the also gamete of heterogametic sex all contains a Y chromosome. Is it correct or not? Is not correct. For example, in GW system, heterogametic sex has the W, but this one has Y, but this one they don't have it. And how about the gamete of the homogametic sex all contains X chromosome? This contains the X chromosome and X chromosome. How about this one? They contain Z chromosome, so it is not right. How about B? Gamete over the heterogametic sex have different sex chromosome, different sex chromosome, and gamete over homogametic sex has the same sex chromosome. So the right one is B. Okay. So in this slide, I will explain the genic sex determining system. In Korean, we call it as 유전적 성결정. So in this system is the no sex chromosome. So only the sex determining gene is existed. This system is found in the yeast and some plants and some fungi and something else. The very famous example is the MATA gene and MAT alpha gene in the yeast. So yeast, they don't have the sex chromosome, but they have the MATA gene and MAT alpha gene. So this two slide comes from my epigenetics 후성 유전체학 course. So in 후성 유전체학 course, the MATA and MAT alpha is very important. Okay, so let's explain it very simple. Okay, for example, if they have MATA gene, for example, they will become the male. If they have the if they have the MAT alpha gene, they will become the female. Okay, but the yeast, they don't have sex chromosome, but they only have sex determining gene MATA and MAT alpha. In previously all over system, they have the sex chromosome. But in the genic sex determining system, they don't have sex chromosome, but they only have sex determining gene. Okay, slide I will explain another sex determination system. One is we call it as an environmental factor. Okay, there are two environmental factors. One is position, the other one is temperature. Okay, how about let's start from the position. So this is limpet. Limpet is the 사과조개. In Korean we call it the 사과조개. 사과조개 is start from the one stack. After that another stack goes in. After another stack goes in, and another stack goes in like this. So okay, this is the stone. Stone is here. At first time, a larvae that settles on the unoccupied space becomes female. So this one, they settles on the unoccupied space becomes female. After that, another will come to here. So this female attract another larvae, and after that another larvae will go to the top of the female. After that, this larvae will become the male. Okay. After that, the male on the top switch sex from male to female. So it means that all 1 and 2 they will become female. After that, they will also attract another larvae. After that, larvae settles on the top of female and becomes male. So it means that limpet position is very important role in sex determination. How about temperature? This phenomenon is happen in the turtle. Okay. So in the higher temperature, all of offspring nearly more than 80% around the when the temperature is around the 29, so about 60% it becomes female. How about 29.5? About the 80%. About the 80% of the offspring will be the female. Okay, when the temperature is very low, around the 26, 100% is the male. How about 28 centigrade? 100% is male. How about 29 centigrade? Only 80% is male, but 20% is female. How about 29.5? About the 20% is male and 80% will become female. This is due to because higher temperature can activate aromatase. Aromatase is the changes testosterone to estrogen. Estrogen is a female hormone. So in higher temperature, around 29.5, they changes testosterone to estrogen. So many offspring will become females like this. So this is we explain the environmental factor. It also can be sex determination. One example is position. The other example was temperature. Okay, in this table it summarized the sex determination systems. We already learned the XO, XX, XO, and XX, XZ, and ZZ, ZW is the sex chromosome is involved in this sex determination step. How about the xenic sex determination? So no distinct sex chromosome is involved in sex determination. How about the environmental sex determination? Usually the environmental factor, one is location and the other one is temperature. And also, how about the heterogametic sex? For example, this is the in XO system, this is heterogametic sex. How about the XXXY? This is heterogametic sex. And ZZ system, ZW is heterogametic sex. And also heterogametic sex is male and females like this, okay? In this slide, I will explain the sex determination system in Drosophila. What is Drosophila? Drosophila is fruit flies. In Korean, we call as chopari. Chopari like this. Chopari also has the female and male. They also have the one pairs of sex chromosome like human, X and Y. And also they have three pairs of autosome. Autosome means that 상염색체. They have sex chromosome means 성염색체. They have three pairs, chromosome 2, chromosome 3, and chromosome 4. So okay, like human, the XX means that they are the female, and XY is also male, like this. For example, the AA means that autosome. They have three pairs of autosome. So AA and XX, so their gamete, the 배우자 will have the XA plus X and A plus X, and in these cases they will have the A plus X and A plus Y. So in this case, when this one and this one is fertilized, they will make the female, and when this one and this one fertilization, they will make the male. Before we know the further, this is very simple. Just looks like the human. But if we see in detail, there is another system. So sex determination in Drosophila is dependent on the X and A ratio. X means that the number of X chromosome. A means that number of haploid set of the autosome. Okay? Okay. So in these cases, what is number of X chromosome? 1 and 2. 2, isn't it? How about the number of haploid set of the autosome? In this case is also 2. So 2 and 2 ratio will be the 1. In this case, at the ratio is 1, it will make the female. How about in these cases, what is the number of the X chromosome? In these male cases, they have only 1. How about the number of the haploid set of the autosome? 1 and 2, they have 2. So the final ratio will be 0.5. It means that it will make the male. Okay. How about in these cases? The sex chromosome is instead of XX, they have the XO. XO means that they only have the one X chromosome. And also the haploid set of the autosome is the A and A. In this case, is also 1, 2, 2 means that 0.5, it will make the male. How about X, X, Y, AA system? What's the number of X chromosome? Is 2. What is number of haploid set of the autosome? 1, 2. They have 2. So it means that 2 means the final ratio will be 1.0. So it means that it will make the female like this. How about in this case is the XXX, the AA. When they have the XXX in the AA, in this case is what's the number of X chromosome? X chromosome will be the 3. What's the number of A chromosome? A chromosome is number will be 2. So final ratio will be 1.5. So it will make, in this case, it will make the meta female. So meta female means that we call it as a super female. It means that their phenotype is female, but the meta female is usually dies very early. Okay, how about in these cases? They have the XXXY, and they have the autosomal set is A and A. What's the number of X chromosome? X chromosome is the 1, 2, 3, 3. How about haploid set of the autosome is 1 and 2. So what's the ratio of X and A? Will be the 1.5. So this one also cases also make the meta female like this. Okay. How about these cases? They have the sex chromosome. They have the XX, and also they have the AAA. AAA. So in this case, what's the number of X chromosome? X chromosome is 2. What is number of the haploid set of the autosome? The number will be 3. The ratio will be 0.67. So 0.67 is located between the 1.0 and 1.5. In this case, we call it as intersex. How about in this case is the X chromosome? They only have the X. X0 means they only have one X chromosome, and their autosome is AAA. So what's the number of X chromosome? X chromosome is 1. What's the number of A chromosome? Is 3. So ratio will be the 0.33. In this case, it's below the 0.5. So we call it as a meta male. Another name is a super male. So in this case, their phenotype is a male, but they die very early. Okay. How about the final cases? XXXX, AAA. So what's the number of X chromosome? Is 4. What's the number of A chromosome? The autosome is 3. So ratio will be 1.3. 1.3 is located above female, so it will make the meta female like this. So important thing is in sex determination in the fruit fly is X and A ratio. So if they are in the 1.0, it will make the female, and 0.5 it will make the male, and above 1.0 it will make the super female, and below 0.5 it will make the super male. And between 0.5 and 1.0 they will make the intersex. Okay. And the concept check. What is the sexual phenotype of fruit flies that has XX, YYY sex chromosome and 2 sets of the autosome? What is the sex phenotype? So what is the number of X chromosome? 1 and 2. What is the number of the haploid autosome? They have 2 sets means that 1 and 2. So it means the ratio will be 2 and 2 means that ratio will be 1. In this case, the sexual phenotype will be the female. Okay. Okay. So in this slide, we will see the sex determination in human. As already you know that our human, we use the XX and XY sex determinations. So usually the Y chromosome is very important role in the maleness. So the reason why the Y chromosome has SRY gene in the upper arm. So this is a sex determining reason why SRY gene. So how do you know that the Y chromosome, especially SRY gene, is very important in the determination of maleness? So this is we can see the importance of the Y chromosome by based on the in the some syndrome. So okay, let's start from the Turner syndrome. So what is Turner syndrome? Their karyotype is that XO, means that they don't have the Y chromosome. Because they don't have a Y chromosome, their phenotype is women, but they don't have a menstruation. How about the Klinefelter syndrome? The Klinefelter syndrome, the karyotype is XXY or the XXXY or XXXY or XXYY. They have the Y. And so their phenotype is that because they have Y, their phenotype is male. But they have the very small testes, and they have a very small guan testes, and they show that infertility. Their phenotype is male, but they are infertility. How about poly-X female syndrome? Their karyotype is the XXX, and you can see that XXX female is the phenotype looks like the female, but they have some menstruation difficulties, but she is fertile and her children is normally have a normal karyotype, but they have the poly-X syndrome. Their phenotype is female because they don't have a Y, but they have some difficulties in menstruation. Okay, based on these findings, we can say that Y chromosome determines maleness because the Klinefelter syndrome, even though they have 2X, or even though they have 3X, even though they have 4X, their phenotype is male. Okay. We will further investigate the role of sex chromosome. So human has the X and Y sex chromosome. Okay. X chromosome contains the genetic information which is essential for both sexes. So at least one copy of X is required. For example, female has XX and the male has the XY because at least one copy of the X is required. The male have the X and Y. And in absence of X is lethal. Lethal means that they dies. So in this case, we can say that because this is Y, the X chromosome contains genetic information essential for both sexes. Okay, how about Y chromosome? In the Y chromosome, the male determining gene, especially SRY gene, is located in the Y gene. So a single Y, even in the presence of several X's, still produces a male phenotype. The very famous example was Klinefelter syndrome. Even though they are XXY, XXXY, XXXYY, their phenotype is still produced male phenotype. But the absence of Y is the Turner syndrome and the poly-X female syndrome result in a female phenotype. So let's summarize again. The X chromosome is the essential for both sexes. At least one copy of X is required. The Y chromosome, they have the male determining gene, especially the SRY gene. So a single Y, even in the presence of several XY, still produces male phenotype. Without the Y, they resulted in female phenotype like this and like this. We will learn especially the male determining gene. Special name is the SRY genes. So SRY gene encodes the transcription factor that stimulate the transcription of other gene, especially what kind of gene? That promotes the differentiation of the testis. So this is testis. Testis is composed of the Sertoli cell, which is very plays important role of the maturation of the sperm. So this is Sertoli cell. So primary spermatocyte located here, and secondary spermatid located here. After that, they will make the sperms like this. And next to the Sertoli cell, there is a radix cell. Okay. So what's the function of the SRY? Once again, it can stimulate the transcription of other genes that promote the differentiation of testis. So, for example, SRY gene is to make the Sertoli cell to secrete MIS. So MIS means the Mullerian inhibiting substance. After that, Mullerian duct degenerated. So this is the Mullerian duct. So usually the Mullerian duct make the ovary. How about the radix cell? So SRY gene can activate radix cell. After that, this radix cell make the testosterone, and usually the testosterone can change it to androgen. After that, androgen can make the Ulpivian duct generation. So this is the blue is Ulpivian duct. So it's very important role the sperm making. Okay, so this is the SRY gene. So again, it's a very important role to promote the genes that plays important role in the differentiation of testis. Okay. In this slide, we will learn the androgen insensitive syndrome. Okay, what is the we call it as AIS. So it means that this is caused by defective androgen receptor. For example, here testosterone is changed to androgen, and androgen can be received by the androgen receptor. So usually the androgen receptor is working very well. After that, it can generate Ulpivian duct like this. This is Ulpivian duct. Okay. We will learn about the androgen insensitive syndrome. Okay. Previous slide, we learned that the androgen, which is changed from the testosterone, is very important role in Ulpivian duct generations. So what is Ulpivian duct? So Ulpivian duct is the man's male 생식기. male genitalia, male 생식기, show that male 생식기, okay? But androgen insensitive syndrome is happened in the male, which has the XY chromosome. So they have the Y chromosome, but they have the defective androgen receptor. It means that even though androgen it can be expressed, but this receptor is not working. It means that they cannot make the ulpivian drug. So that's why, so this is the image 2. So this is the newborn and genetic male. They have the X and Y, they have a male, but they have the androgen insensitive syndrome, and they have the female external genitalia, female 외부 생식기를 가지고 있다. And also they look like the women, so possibly feminized picture. So even though they are X and Y, even though they can make the androgen, but they don't have the androgen receptor, so they cannot make the ulpivian drug. So their phenotype looks like women, and they have the female external genitalia. So based on this finding, what we can say? We can say that here the Y chromosome is the SRY gene is very important role in the maleness, and also XRY gene, okay, they can stimulate the transcription of other gene. What kind of other gene? That promote the differentiation of the testes. So usually the SRY gene can activate the production of testosterone, and also testosterone is changes to androgen. But also, if you want to make the ulpivian duct generation, the androgen receptor is also very important. So in the case of AIS, androgen insensitive syndrome, so they have, even though they have XY, but their phenotype looks like the female, like this, okay? We will concept check once again. What is the phenotype of person who has a XXXY sex chromosome? So they have the Y chromosome. We can say that if they have a Y chromosome, they have a SRY gene. If that SRY gene can activate the production of testosterone, and testosterone can be changed to androgen, and androgen plays important role generation of ulpivian drugs. So their phenotype will be male, and also we will call it as the Klinefelter syndrome. Okay. So in this slide, from this slide, we'll study the second subject, sex-linked characteristics. Okay, what is sex-linked characteristics? Okay, before that, we can summarize the non-sex chromosome and sex chromosome is again. What is non-sex chromosome? In other words, we call it as the autosome. Our human has the 22 pairs of autosome, start from chromosome 1 to chromosome 22. And also we have the sex chromosome. For example, men has XY, but women has XX. So what kind of gene is located in the autosome? There are a lot of gene is located in the autosome. But how about the Mendel's gene, which encodes the seed color and seed shape and seed coat color, and also pod shape, pod color and pod shape and stem length? All of all of this kind of gene is located in the autosome, located in the non-sex chromosome. But Morgan, 19,000, early 19,000, Morgan found that the gene which encodes the color of the fruit flies is located in sex chromosome, especially in X chromosome. So this is, we call it as sex-linked characteristics. Okay? This slide we will learn the X-linked characteristics. So Morgan found that the locus affecting eye color in Drosophila is located in the X chromosome. And somehow he also found that white eye is the recessive trait. So red eye is dominant than the white eyes. Okay, the question was that are the white eye in fruit flies inherited as an autosomal recessive trait? So what is autosomal recessive trait? So we already learned that the gene which encodes the Mendel's phenotype is located in the autosome, and the large A is dominant than small a. Small a is recessive. So if they are located in the autosome, so this is a small a phenotype, so they are recessive. So this is a recessive phenotype, but after crosses F1 generation, they will show the dominant phenotype because the genotype is a large A, small a. So they show that all of them, they will show the dominant phenotype. But F2 generation, the ratio 3:1, they will show the recessive phenotype. So this is the autosomal recessive trait. If the gene which encodes the eye color in Drosophila is the recessive, in F1 generation, we cannot see the eye color. And F2 generation, we will see the ratio will be 3:1, but he found that the ratio is different. And also this is another case. So this case is the white-eyed male cases, and this case is white-eyed female cases. So if this gene which encodes the white gene is located in the autosome, in F1 generation, they will not show the recessive phenotype, but in the F1 generation, they found that recessive phenotype in the F1 generation. And also, in the F2 generation, the ratio is totally different than 3:1. That's why Morgan thinks that maybe this gene which encodes white eye is located in the X chromosome. Okay? For example, because the W is recessive, then the plus means that red eyes. Okay, so he used the white-eyed male, so genotype with the XWY like this, and also he crossed with the wild type X plus X plus. So usually the wild type like this, crossed with wild type. Okay, and also in these cases when the male was the white eyes, so gamete will be this one. Female make the X plus as a gamete, and also white-eyed male, they can make the XW and or Y's like this. X fertilization, they will make the X plus XW or X plus Y's like this. all of the female show that red eye, and all of male also show the red eyes. After that, they did fertilization again. They used their F1 generation, male and female, and the female, they will make the X plus the XW gamete, and also red-eyed male, they will make the X plus or Y. After fertilization, so this is Punnett scale. So X plus, this comes from sperm. Sperm is X plus the Y, and the egg is X plus XW. So in this scale is the X plus means that they are red-eyed female. So how about this one? This one is X plus Y, and they show that the phenotype is red-eyed male. How about this one? This one is X plus XW, because W is the recessive then plus, and they will show that red-eyed female. How about this one? They will make the XWY. Finally, they will make the white-eyed female like this. So the ratio is that the red-eyed female and red-eyed female means that the 2 out of 4 show that red-eyed female, and also 1 out of 4 show that red-eyed male, and also 1 out of 4 show the white-eyed male like this. So ratio is a little bit different than 3 to 1. Okay. How about in the other cases that the female show that the recessive, the white-eyed female, so the genotype will be the XW and XW. And they cross with the wild-type male. So wild-type male means that X plus Y. So in these cases, they will make the XW as a gamete. And for example, red-eyed male, they will make the X plus or Y. So after fertilization, all of the, because this one is crossed with, when this one is crossed this one, all of the females show that red eyes. But how about the male? All of the males show that white eyes. So after that, they did self-fertilization. So this comes from the male sperm. So one is plus XW and Y, and this is they will make the egg X plus XW. So this is Punnett scales. So in this scale, they will show that X plus XW means that red-eyed female. How about this one? This one is X plus Y. They will show the red-eyed male. How about this one? They will show that XW means that they will show the white eyes. How about this one? They will also show the white-eyed male. So final ratio was the 1 out of 4 will show that red-eyed female, and 1 out of 4 show the white-eyed female like this, and also 1 out of 4 is red-eyed male, and 1 out of 4 is white-eyed males like this. The question was that are the white eyes in the fruit-wise inherited as an autosomal recessive trait? No. The result of reciprocal crosses are consistent with X-linked inheritance. So they are not autosomal, they are sex chromosomal. Also, white eye is a recessive trait. If they are autosomal recessive trait, their F2 generation, their phenotype will be the 3-to-1, but their phenotype is totally different. It means that they say that it means that they are not autosomal, but they are in X-linked. In this slide, I will describe some characteristics in the X chromosome. So this is the Drospella's chromosome. So as I mentioned earlier, the Drospella has the sex chromosome. XX is female and XY is male. And they also have 3 pairs of autosome. So one is chromosome 2, and chromosome 3, and chromosome 4. Chromosome 2, and chromosome 3, and chromosome 4. Usually the non-disjunction usually happen in the X chromosome. For example, XX is female, so when they make the gamete, usually the X can go to the here and X can go to the here. But usually 10% of cases, non-disjunction usually happen. So one gamete becomes the XX, the other gamete, they will have no X chromosome. So this is the sperm. So sperm one has X, the other has Y. So this is a Punnett scale. So after this one and this one is fertilized, they will make the X, X, X. So it means that they will make the meta-female. How about this one? This one, after this one and this one is fertilized, they will make the X. So in this case is XO, means that they will make the male. How about these cases? They will make this one and this one fertilized, they will make the X, X, Y. So X, X, Y, they will make the female. How about this one? This one and this one fertilized and they will make the Y. So Y cannot survive. Okay, so in the X chromosome, non-disjunction is happen nearly 10%. Okay, but sometimes in autosome is the non-disjunction happen, but the case is very low, less than 1%. Okay, so this is one example which has the white eyes, but they have the XXY. Because the white eye is recessive, the genotype will be the XW, XW, and Y. So usually 90% they will make the one X and one Y, and finally they will make another XW like this. But sometimes 10% of times they will make the one gamete, they will make the XW, XW, and the other gamete, they will make the Ys like this. So this is some characteristics of X chromosome in Drosophila. Okay, non-disjunction happens around 10%. Okay, in this slide I will explain how people found the gene for white eyes is located on X chromosome. Okay, let's go back to the previous slide. So, based on this result, they found that white eye in fruit flies is not inherited as an autosomal recessive trait. So it means that the gene which encodes the eye color is not located in the autosome. Because if they are located in the autosome, in F2 generation, the ratio will be 3-to-1. But in F2 generation, the ratio is different. But based on this result, we cannot say that the white eyes the gene which encodes height-wise is located in the X chromosome, because there is also another sex chromosome, Y chromosome is also existed. Okay, based on this result, we cannot say, but in this slide, we know that some characteristics of X chromosome, usually in X chromosome, 10%, in 10% non-disjunction is happens. Okay. Okay. So okay, this is the one experiment to show that the question was the in a cross between the white-eyed female and the red-eyed male, and they found that a few white-eyed female and also a few red-eyed male. Why this kind of things happen? Okay, because so usually the white-eyed female, the female is, you can see that this one is XX, or this one XXY is female. So if they are XW, XW, XX, so in the after crosses, all of F1 generation, half of them show that red-eyed female, and half of them they will show that white-eyed male. But they found that the several different ratio. Some of them is show that 1 to something. They have some different ratio like this. So it means that white-eyed female is not XW, maybe XWY. Okay. Okay. So this is a white-eyed female. Okay. So they cross with the red-eyed male. So this is X plus Y. So usually when the separation happens, usually the gamete from this one will become the XWY and XW. After that, so this one they will make the usually the XWY and XW. After that, this male, the sperm, they will have the X plus and Y. After the crosses, they will, in this scale, is X plus XWY. It means that this one is a red-eyed female. How about these cases? They have the XWYY, means that they are the white-eyed, the male. How about this one? They will have the X plus XW. This one will make the red-eyed female. How about this one? They will make the, based on this Punnett scale, they will make the XWY. It means that white-eyed male. Okay, so it means that all of F1 generation will be the red-eyed female and the red-eyed female, like this. And also this one is also white-eyed male, white-eyed male. But so usually this phenomenon is nearly similar to this one. But they found that a few new white-eyed female and also red-eyed male. So based on this one, all of the red-eyed is female, but they found that red-eyed male, and also they found the white-eyed female. Why this is happen? because the non-disjunction is happen. If not, usually the 10% of times non-disjunction is happen. So in these cases, they will make the one is XW and XW, and one is they can make the Y. So when they make the gamete like this, and also this one, red-eyed male, they will make the X plus and Y. So this is Punnett scale. Okay, in these cases, they will make the X plus XW, XW. In these cases, what's the phenotype? Because they have the plus, their eyes will be the red, and this is the XXX. XXX is a meta female. We call it a super female, but they easily die. They usually die. So they usually die like this. How about this one? This one is they will make the XW, XW, Y. It means that they will make the white-eyed female. Usually all of them should be male, but they also found that white-eyed female like this. So this progeny is resulting from non-disjunction. How about this one? They will make the X plus Y. So it means that the eye color will be the red, and so red-eyed male. When this one, they fertilize, they will make Y and Y. Because they don't have X chromosome, they will die like this. So the reason why people can find a few, usually in F1 generation, when the white-eyed female and red-eyed male was crossed, in both cases, usually all of the female is red-eyed, and also all of male will be the white-eyed. But sometimes they found that this one. In this case is also when the separation happens, so usually all of the female is red-eyed, but all of the male is white-eyed. But they found that this phenotype and this phenotype, it means that this phenotype and this phenotype comes from non-disjunction from the XW, XWY. Usually this one happens 10% of times, like this. Okay, so the conclusion is that the white-eyed female and the red-eyed male in the F1 generation is a result from non-disjunction of the X chromosome in XXY female. Okay, so this case comes from the result from non-disjunction of the X chromosomes in XXY female. Okay, so this is concept check. So what was the genotype of the pure live F1 red-eyed male? So this is the pure live, the F1 red-eyed male obtained by Bridgie. Who was Bridgie? So the Bridgie, the scientist Bridgie crosses. So he did crosses and he proved that gene based on this, the non-disjunction. So he found that maybe gene for white eye is located on the X chromosome because the non-disjunction, because this one only comes from non-disjunction, but usually in X chromosome, 10% usually in 10% non-disjunction is happen in X chromosome. So that's why he finally found that he proved that the gene for white eye is located on X chromosome. So scientist Bridgie. Okay, so Bridgie, when he crossed with a white-eyed female with red-eyed male. So what's the genotype? Genotype will be this one and this one. So maybe this one is a X plus Y. So maybe the solution was, the answer was X plus Y. It's like this, okay? Okay. In this slide, I will explain another X-linked characteristics. So usually the color blindness is X-linked characteristics. For example, can you see what can you see inside the circle? If you can see 12, it means that you are not color blindness. Okay, so in the classroom, maybe none of them is color blindness. Maybe if there is 1,000 people, maybe one or two people will have the color blindness. It means that color blindness is recessive trait. Okay, color blindness is a recessive trait. Okay, so if they are recessive trait, okay, they are recessive trait, and also if they are located in the autosome, not X chromosome, in F1 generation, all of them will show will not show the color blindness. Okay, but we found that it's okay. Okay, when they cross with the normal female and the colorblind male, okay, so this is the normal female, and normal female is the X plus and X plus, and colorblind male will be the X, C, and Y, like this. In the gamete, in the egg, they will make the only X, and the male in the sperm, they will make the X, C, and Y. So in this Punnett scale, they will make the X plus XC, means that normal color vision female. How about this one? In this scale, they will make the X plus Y. This means that they are also normal color vision male. Based on this F1 generation, we can say that maybe if the gene which encodes for color blindness, if they are located in the autosome, maybe it can be located in the autosome or the X chromosome. But okay, when the parent generation, the female was colorblind, so means the XC and XC, and the male was normal plus X plus and Y. So in this case, they will make the gamete like the X 배우자 XC XC. So the egg will be the XC, and sperm they will make the X plus and Y. In this case, they will make the X plus XC, means that they will have normal color vision female. But in this case, they will make the XCY. It means that they will make the color blind male. Based on this result, we can say that this is not, or this gene which encodes for color blindness is not located in the autosome, so it's located in the X chromosome. Okay. Okay. Next one is that, okay, this is the one question. So Betty has normal vision, but her mother is colorblind. So Bill is colorblind. Okay. So if Bill and Betty marry and have a child together, what is the probability that the child will be colorblind? Okay, so Betty is normal vision, but her mother is colorblind. So it means that Betty is carrier. Okay, so based on this one. So Berry is the carrier X. They have her genotype is X plus and XC. How about Bill? Bill is colorblind, means that his genotype is XCY. So based on the Berry make the gamete like X plus the XC. How about Bill? Bill make the gamete maybe X, C, and Y. So this is they will do the fertilization. So this is Punnett scale. So Punnett sperm, they will make the X, C, and Y. The egg, they will make the X plus and X, C. So in this scale, they will make the X plus X, C, means that they will have the normal vision female. How about this one? They will make the X, C, and X, C, means that color blind the female. How about this one? They will make the X plus Y, means that normal vision male. How about this one? They will make the X, C, Y. In these cases, they will have the color blind male. So what is the probability that the child will be colorblind? The total will be the 4. Among 4, 2 will have the colorblind. So it means that 1 out of 2. Okay. So this slide is concept check 5. So hemophilia. Hemophilia is the reduced blood clotting. Is an X-linked recessive disease in human. A woman with hemophilia mates with a man who exhibits normal blood clotting. What is the probability that their child will have hemophilia? Okay. How about the women? So women, the genotype will be the XH and XH. How about the male? Male shows that normal blood clotting. They will have X plus and Y. So in the Punnett scale, the female make the egg and XH. How about male? In sperm, they will make the X plus and Y. In the Punnett scale, so this one, they will make the X plus XH. This one, they will have female with normal blood clotting. How about this one? They will have XHY. It means that this is male who will have the hemophilia, reduced blood clotting. So what's the probability that their child will have hemophilia is 50%. Okay. In this slide, we will see the Z-linked characteristics. As we mentioned earlier, Z-linked characteristics is shown in reptile and some birds. Okay. In this case, Z is male and ZW is female. So, in the camel color, in the Indian people, what is Indian people? People is 공작. 한국말로 공작이죠. Camel color looks like the brown color. Camel color is inherited as a Z-linked recessive trait. This is a recessive trait because most of the Indian people is blue color like this, blue. But some of them, nearly less than 10%, they will show the camel color. It means that camel color is a recessive trait. Okay, so first case is this one. So parent generation, blue female, and the male was camel color. Because the camel color is the Z-linked recessive trait. The genotype will be this one, will be ZCA and ZCA. How about the female? Blue female. Blue female will be the ZCA plus and W, like this. So after meiosis, this one make the egg, one egg is like the ZCA plus, the other egg is W. This one, they will make a sperm. All of sperm will have ZCA, like this. So after fertilization, fertilization happens. So this is F1 generation. In this care, we see that ZCA plus and ZCA. It means that Z means that this one is male, but the color is, because this is the dominant than this one, color is blue male, like this. How about this one? In this care will be the ZCA and W, means that this one will have the ZW will be the female, and the color will be the camel female. So after meiosis, this male make the ZCA plus, this one ZCA. How about the female? Female make the ZCA and W. So this is a Punnett scale, okay? Punnett scale like this in F2 generation. So sperm will be the ZCA plus and ZCA, like this, okay? How about egg? Egg will have ZCA or W. So in this scale, they will have ZCA plus ZCA, means that the color, this Z means is male and color is blue. How about this one? This one, they will have a ZCA plus and W, means that ZW means that female, and they will have the blue color. How about this scale? They will have ZCA and ZCA, means that Z means that male, and color is camel male, like this, okay? How about this one? This one is a ZCA and W, means that this is the camel female. So ratio between the blue male and the camel male and the blue female and the camel female is 1 to 1 to 1 to 1. How about the reciprocal crosses? Reciprocal crosses means that the female is the camel color and the male is blue. Previously, the female was blue and male was camel. In this time, female is the camel and male is blue. Okay. So after meiosis, in parent generation, this female make ZCA and W, and this male, they will make all of them ZCA plus. So after fertilization, so the sperm can go to here, and egg can go to here and here. So this is Punnett scale. So in this scale is ZCA plus and ZCA, means that Z means the male, the color is blue. How about this one? ZCA plus and W, so it means that the color is blue. So all of offspring will show that the blue color. So in the F1 generation, after meiosis, this male make the sperm ZCA plus and ZCA. How about this female? This female make the egg. One is ZCA plus and W. So in this care, after fertilization, this one will make the ZCA plus and ZCA plus. It means that ZZ will have the male and the color is blue. How about this one? This one is a ZCA plus and W. ZW is female. The color is because they have a Ca plus, color is blue. How about this one? ZCA plus and ZCA, the color is blue and the sex is male. How about this one? This one is that ZCA and W means that color is camel and sex is female. So the ratio between blue male and blue female and the camel female is 2-1-1. Okay, because the camel color in the 공작새의 camel color is inherited as a Z-linked recessive trait. Okay. In this slide, we will learn the Y-linked characteristics. Previous slide, we learned about Z-linked characteristics and also learned about X-linked characteristics. Okay, in this slide, we will learn the Y-linked characteristics. Y-linked characteristics is only present in the male. It's only present in the male. Because the Y chromosome is only present in the male. If the father was affected, all male offsprings will exhibit the traits. They will be affected like this. So example is a flat foot, 평발, and also hair loss in male. So this is the memory. So this is the Y-linked characteristics. Okay. In this slide, we will learn the dosage compensation. So dosage compensation is also X-linked characteristics. So dosage compensation means that equalized the amount of protein produced by a single X and two autosomes in the heterogametic sex. For example, what is heterogametic sex? For example, male is in human, male is X and Y. Because they have X and Y, so we call it as heterogametic sex. How about Drosophila? Drosophila is also X, Y is the male. It's okay. So to equalize the amount of protein produced by a single X, they have two different mechanisms in Drosophila and mammalian. For example, Drosophila, the male is X and Y. So in Drosophila, they doubling the activity of genes on the X chromosome in males because they only have one X. So Drosophila is doubling the activity of one X chromosome. So it means that the one X chromosome is function as 2X. How about mammals? Mammalian is that mammalian female has 2X, but they randomly inactivate one of X chromosome in female. So randomly, sometimes or this one randomly inactivate this one, Okay, only this one is functional. How about in the sometimes one only this one is inactivated, only this one is function. So this is mammalian is just the female, they have two X, but they randomly inactivate the one X chromosome. This is the mammalians. So this is the mammalian cell. Okay, this is nucleus from the female, and this is nuclear from the male. So this is nucleus. So in the edge of the nucleus, so this is the nuclear membrane is located here. So near the nuclear membrane, there is a condensed in the darkly staining bodies, like this one. So this is the bar body. So the scientist Ryan found that this bar body is an inactive X chromosome. So all of inactive X chromosome they are located here in the edge of the nucleus. So the shape is condensed and darkly staining. For example, this is table 4.3 to show the number of bar body in the human cells with a different complement of sex chromosome. For example, female has XX. In this case, number of bar body is 1. How about male? XY. They don't have a bar body. How about Turner syndrome? They only have the 1X. They don't have a bar body. How about Klinefelter syndrome? XXY. In these cases, they randomly inactivate the X, so they will have the 1. How about XXYY? They randomly inactivate the 1X, so they will have 1. How about XXXY? They will randomly inactivate the 2 X chromosome, so they will have 2. Yeah, something like that. Okay. So in this slide, we will also learn about the dosage compensation. So the one of the X chromosome is randomly inactivated. So random inactivation. So this is cat. The cat name is the tortoise cell cat. This cat. The coat color is some of the region is brown, and some of them is the black like this. So this is the patchy distribution. This is patch distribution. But the patch distribution of color in the cat is a result from the random inactivation of one X chromosome in the female. So this cat is female, okay. So this is usually the X chromosome is the one comes from father, one comes from the mother. So they will have usually the cell has the 2 X chromosome. So during the mitosis, so after mitosis happens, one cell becomes the 2 cell. So this is the cells. So after 2 successive mitosis, one cell becomes 2 and 2 cell becomes 4. This is 4 cell. Okay, after that, in this case, this is in this cell, the X chromosome comes from mother is randomly inactivated. This cell is also X chromosome from mother is randomly inactivated. And this chromosome in this cell, the X chromosome comes from father is randomly inactivated. The father is randomly inactivated. So this cell also did mitosis, and one cell become 2, one cell become 2, and one cell become 2, and one cell become 2. So in this quote, some cells have only uh, only this kind of cell. For example, the black color is comes from this one, and brown color is comes from this one. That's why they have the patch distribution of color. So this is the due to from the random inactivation of one X chromosome in the female. This kind of coat color is only happen in the female. Okay. Okay. So okay, in this chapter we learned about sex determination. First we learned about sex determination. Second we learned about sex-linked characteristics. Finally we learned dosage compensation. Okay, thank you. Today we will finish the class. Thank you.